Hey guys, this is Mr. Mahmood. This lecture will be to introduce some of the more advanced genetics. In this case, we're going to talk about two different types of dominance called incomplete and codominance. Let's get a little more complex now. Up to this point, all we talked about was either showing for the dominant or the recessive trait. Now we're going to talk about two different possibilities. One is something called incomplete dominance. The other is called codominance. Now they both pretty much represent what you would think from looking at the term. Incomplete dominance means it's not fully dominant. Right? So in this case, again, we'll stick with certain alleles. So we'll stay with the tall and short scenario. Let's say again the capital T represents tall, the lowercase t represents short. But now, homozygous and heterozygous tall are not going to be the same. So here's the basic scenario. If I have someone who's homozygous dominant, that means both of their T's are capital, right? And then I have someone who's heterozygous for the same trait, one capital, one lowercase. The third possibility is homozygous recessive. Now the first two would be exactly what you would expect. This person would be tall, or this plant, would be tall. This plant would be short. However, this one, instead of having that same dominant trait that you would expect to have, as long as you have at least one dominant allele, if we're talking about something as being incompletely dominant, and the question will always tell you, I'll always say this represents incomplete dominance. If using incomplete dominance, what would be the probability of certain things happening? If you have this particular genotype, it actually represents its own third phenotype. So in this case, the third phenotype would be some sort of combination of the two, or we'll call this like an average height. So if there's an average height plant, that one has what's considered incomplete dominance for the trait. They have one dominant, one recessive allele, so it's incompletely dominant. Another good example, if you think colors, again this is an oversimplification, but let's say for a given plant petal, the dominant color was red, the recessive color was white. So if we dealt with complete dominance, that's what we were dealing with before, as long as you had at least one dominant allele, you always showed for the red plant or the red petals. But in this case, if it was incompletely dominant, someone who was heterozygous would be a mix of the two. So anytime you're dealing with a mixing of two possible phenotypes to produce a new third phenotype, we call that incomplete dominance. So in this case, instead of red and white, you would have a mix of the two, so something like a pink offspring. So a pink flower being produced from a red and white flower is a good example of incomplete dominance. And so if we're dealing with incomplete dominance, heterozygous is actually its own phenotype. And again, this is a very rare situation. Never assume this. Always just assume at least one dominant allele shows for the dominant trait. But if you're told use incomplete dominance, then you understand heterozygous is the only difference here. It actually has its own phenotype, and it's usually a mix between whatever the dominant and recessive phenotype would be. We're not even going to worry about doing crosses here representing it because it's the same crosses, except in this case our heterozygous genotype now has its own phenotype. Now let's focus in on codominance. Codominance means that there's actually more than one dominant allele. So this is going to get a little tricky because we're dealing with the same trait. So we're not going to deal with a different letter, but there are going to be different possibilities of a given letter in terms of the dominant allele or the dominant trait. A perfect example of codominance in the human body is sickle cell anemia. Sickle cell anemia is a disorder of your red blood cells. We've talked about red blood cells before, how amazing I think they are and everything like that. But red blood cells are typically circular and they carry oxygen along with other things to the rest of your body. They're basically your body's highway, and they're what carry things throughout your body. If you have sickle cell anemia, that means that your red blood cells aren't all perfectly round, but they actually now have sickle shapes, which means they're not that perfect round circle, and that causes problems. One, they can't carry as much oxygen, and two, they can get in each other's way a lot more easily. They can tend to block blood vessels, cause blockages in the body, which could become pretty serious as you get older. So sickle cell anemia is not necessarily a good thing to have. It's considered a codominant trait. So of the possible alleles, instead of thinking as one dominant and one recessive, both of the alleles that we deal with in terms of sickle cell are considered dominant. So instead of there being a dominant and recessive, we use two examples of dominant. For whatever reason, we use in genetics the letter N as the typical representation of sickle cell anemia for this particular trait. So that gene is particularly used as the letter N is represented here. Now, the capital N is true for both possibilities. So the difference between these two dominant alleles is that one of the N's is considered normal, and that one's usually represented with a dominant A superscript, and then the other N is considered the sickle shape, which is a capital S superscript. So these are both actually dominant traits. there is no recessive allele for this particular situation. So the Na represents normal red blood cells. I'm just going to write RBCs here. And the S represents your sickled RBCs. And sickled are the ones that are irregularly shaped that can cause problems. Now, most of the human population is homozygous Na. That means most of the human population has 2 dominant NAs, which means 100% of our red blood cells are normal. But there are certain groups of the population that are either heterozygous for this trait, 1 dominant Na with 1 dominant NS, or homozygous NS in this case, meaning 100% of their blood cells are sickle shaped. Now it's the combination of the two that's going to be unique. So the NS is no surprise. 100% of their red blood cells will be all sickle shaped, which is a big problem. It means they're constantly having trouble getting oxygen to their cells, multiple problems with blockages throughout their body. However, it's this middle variety that distinguishes codominance from incomplete dominance. If you had one of each, if we're talking about incomplete dominance, there'd be some sort of mix between the two, but that's not the case here. This is what we call codominance. So instead of a mixing, each individual trait is represented, but both of the dominant alleles are represented at the same time. So this means of the 100% of your red blood cells, someone who's this middle characteristic of 1 dominant Na and 1 dominant NS will have about half of their red blood cells be normal and about half of their red blood cells be sickle shaped. So the half that are normal are just like anyone else's, and there's absolutely no difference between them. But the other half are completely sickle shaped. So this is a good representation of codominance. So let's do a basic cross here. Let's say I cross someone who is 100% normal, homozygous normal, with a homozygous sickle individual. So in this case, I have my 2 alleles, not just one letter, but the letter with the superscript being represented. So the first parent's homozygous normal, the second parent's homozygous sickle. When you do the cross, what do you expect to happen? You're going to get 100% offspring that are codominant for the trait, meaning about half of their alleles are going to be representing the normal red blood cells and half are going to be representing the sickle. So this individual will have sickle cell anemia, but they will have healthy red blood cells as well. So they'll be able to live a much more healthy life than someone who's in this bottom situation where 100% of the red blood cells are sickle shaped. So you'll learn a bit more about this in your classrooms, but this is just a basic example of codominance. The major example of codominance that we talk about at this point deal with blood types. Now I think you guys all have some familiarity with blood types. If you don't know your own blood type, I strongly encourage you to pause this video right now, go ask your parents what your blood type is. If they don't know, I encourage you to try to figure it out. If you've ever had your blood drawn, if you've ever had a doctor's visit with a normal routine checkup or blood work done, someone knows your blood type. It's good to know what your blood type is and keep it on you at all times, just in case something were to happen and you needed a blood transfusion or something like that. They could find that information quickly. Now, you can fall into one of 4 human blood types. There are 4 basic categories of blood types, and they all have to do with a very particular marker protein that's found on your red blood cells. You see how all this is coming together. Think back to what you knew about cell membranes and cell transport. There are 3 types of proteins on the surface of a cell membrane. One of those are your marker proteins. Your marker proteins are responsible for identifying the cell to the outside environment. In this case, there are different categories of marker proteins on a human red blood cell, and depending on which combinations of these proteins you have, that determines your blood type. For example, the first blood type is what we call type A. This individual has the A marker protein. We also refer to this as an antigen. An antigen is any kind of a surface protein. So antigen is a general term for all proteins on the cell membrane. So we're going to refer to the A antigen here as the A marker protein. So if you have type A blood, that means all of your red blood cells have the A marker protein on the surface of your cells, of those red blood cells. If you have the type B blood, that means you have the B antigen or the B marker protein on the surface of your red blood cells. You can also be type AB. If you're type AB, this is the example of the codominant trait. Here you're noticing 2 possibilities, both independently represented. They both are represented on the surface of your red blood cells. If you're type AB, that means you have the A and the B antigen on your red blood cells. There's not some sort of mixing of these 2 to make a new type C marker protein or anything like that. Each one is independently represented. It's the exact same A antigen and the exact same B antigen. You just happen to have them both on your red blood cells instead of just one. And then finally, you can be type O, which means you have neither of the 2 marker proteins on your red blood cells. So this individual doesn't have the A or the B antigen on the red blood cells, which means for some very specific things because of it. So here's the whole idea. I think you guys all know that you can't just mix any blood type with any blood type. They're very specific about which ones can be mixed with others. It's all because of these antigens. It's all because of this right here. You're going to learn about how your immune system works fairly soon. And basically your immune system learns to fight things that it's not used to. And that's how your body's developed its natural immunity. It's how the human species has survived as long as it has. If your immune cells ever see marker proteins that it's not used to seeing, it basically treats it like a threat, and it attacks those cells, thinking that those cells could be harmful, like a virus or bacteria, some sort of pathogen or disease-causing agent. And it's let your human species survive for quite a long time. Any sort of disease or infection that gets into your body, your immune system can pick up that it's not supposed to be there because its marker proteins are unique, and therefore it attacks it. And that's how we survived for as long as we have. Now, unfortunately, modern medicine has began the process of actually purposely putting things in your body that aren't supposed to be there, but with an attempt to try to help you. So when we're purposely trying to put things into our body, even though we're meant to help, your immune system, if it sees it as a threat, it sees something as it hasn't seen before, it attacks it, which is a bad thing. So blood is a perfect example. Only in the past 100 years or so have we purposely put another person's blood into someone's body because we need them to try to regain their consciousness. Let's say they lost a lot of blood for an accident or something like that. That's been pretty recent, and evolution hasn't really caught up to this yet. So your immune cells don't quite understand that blood coming into your body is always meant to help, and 99% of the time it is. So your immune cells basically just see any sort of a marker protein that it's not used to as a threat and attacks it. So we as scientists and as doctors and as people in the medical field have to be aware of this, and we're very careful about which blood types we give to other people, because we need to make sure that we can basically trick our immune system. We want our immune system to not see the blood that we're putting in as a threat. And basically the only way to do that is to make sure you're not introducing any new marker proteins that that person's not used to seeing. So here's how it all works. Your body, your immune system, develops something called antibodies against things that it's meant to attack. So for example, if you have the type A antigen, you have the A marker protein or type A blood, that means you have the anti-B antibody. This means that you as a type A blood will attack anything that has the type B antigen, because your immune system only recognizes what is normally found in your body. If you're type A blood, you only have the A antigen, you don't have any Bs. So you develop the anti-B antibody because you're going to attack anything with a B. So if you have type A blood and somebody with a B marker protein's blood comes into you, your body will attack it. And attacking red blood cells means clotting and what's called coagulation of those dead blood cells, and that big clotting can cause severe blockages in your blood vessels. Depending on how much blood gets transfused, it can be severe issues in terms of organ failure and organ system failure, and it could be fatal. You could die from a blood transfusion of two blood types that don't work out together. This happens occasionally because doctors and nursing staff and people miscommunicate. They get the wrong blood mixed together, and when you're talking about organ donors and things like that, everything has to match, including blood types. So if the blood types don't match, then you have serious problems because of the blockages. Basically, the immune cells are attacking those cells because their antibodies recognize the antigens that aren't supposed to be there. So if you're type A blood, that means you have the anti-B antibody, so you'll attack anything with a B antigen. If you're type B blood, you have the B antigen, which means your body is used to the B antigen. What it's not used to and what it's developed an immunity against is the A. So if you're type B blood and the A antigen is brought into your body, you're going to attack it. And so we have to make sure that doesn't happen. If you're type AB blood, that means you have both antigens. That means that your body is used to seeing both the A and the B marker proteins. Both antigens are perfectly okay for someone with type AB blood. So in this case, they won't develop either of those two antibodies since they're both already present in the body. Right? So someone with type AB blood has no threat by blood types coming in with any combination of the A or the B antigen because it's already present. Their immune system already sees it all the time, and so they just recognize it as part of their own body. So they just recognize it as part of their own blood cells. And then finally, if you're type O blood, you don't have either antigen. So guess what that means? You develop both the anti-A, and the anti-B antibodies. You will attack any combination of the A or the B antigen because they're completely unique to anything that your body normally has. So because of this, we now understand which blood types can mix with others. Alright, so now let's see how we can mix it together. I'm going to fix this. Instead of saying can share with, I'm going to change this to can receive from. We can talk about who you can share with as well, but I think it'll make more sense if we just focus on you as an individual who you can get your blood from. Okay, so let's talk about who each of these can receive their own blood from. If you're type A blood and you need a blood transfusion, you can receive blood from any blood type that does not have a B antigen because you will attack the B. You already have the A, so you're not going to attack any combination of A, but you will attack any form of the B antigen that comes in. So that means you can receive from anyone that does not have the B antigen. So you can receive from yourself. You can always receive from your own blood type. If you're type A, you can receive from yourself. And then you can receive from anything that doesn't have the B. Notice how both the B and the AB, they both have the B antigen. So I can't receive from either of these two. But because type O doesn't have any antigens at all, it won't pose any sort of a threat to a person with type A blood. Because all I'm worried about is the B. As long as the B is not there, I'm not going to attack it. All right? So type A blood can receive from itself or type O. If you're type B blood, that means you can receive from anyone without an A. Because if the A antigen is introduced to you as a B type blood, you're going to attack it. So that means you can receive from anything without the A. So you can receive from, again, yourself, but you can't receive from A or from AB because they both have the A antigen. So the only other blood type you can receive from is type O, again, because it doesn't have either one of those two antigens, so it doesn't pose any kind of a threat. If you're type AB blood, because you have both antigens, you don't have the antibodies against either one of them, or else you'd start attacking your own blood cells. So that means you won't attack any combination of A or B, or any combination of these antigens at all, which means you can receive from any of the four. You can receive from yourself, but you can also receive from A, because you have the A antigen already. You can receive from B, because again, you already have the B antigen. You can receive from yourself, or you can receive from O, again, because O doesn't introduce any new threats to your bloodstream. So type AB blood can actually receive from anybody. Whereas type O, because it doesn't have either one of the antigens, it'll attack any combination of A or B. So of the four blood types, the only one that doesn't have either of the two antigens is itself. So if you have type O blood, you can only receive from other people who have type O blood. And this is why type O blood is such a high commodity. And when we talk about the clotting factors, that's basically what we don't want to happen. If you have type A blood, you're going to clot with any one that introduces the B antigen, which means you're going to clot with either type B or type AB. If you have type B blood, you're going to clot with any combination of A, which means either the A or the AB will cause clotting factors. These are things you try to avoid at all costs. If you're type AB blood, because you have both antigens and neither one of the antibodies, there isn't a blood type that will cause a problem for someone who's type AB blood. And then finally, if you're type O, that means you have both antibodies already. You're going to clot with any combination of A or B. So you can't actually combine with any of the other three blood types. You can only receive from yourself. So this is where we get into the idea of what's considered the universal donor and the universal receiver or recipient. The universal recipient, we'll call UR, is what we consider type AB blood. If you have type AB blood, you can receive from just about any of the four blood types because you already have all the antigens. You're not going to develop the antibodies against it. Now on the other side, the universal donor, UD, is what we consider type O. If you're type O blood, that means you can give to any blood type because you're not going to introduce any new antigens of those two because you don't have either one of them. So type O blood isn't a threat for other blood types. But if you yourself are type O, you can only receive from other type O blood. That's why people who are type O blood are encouraged to donate as much as possible. All right, so there are exceptions. There's one more variable that we play in here, which we consider the rhesus factor. This makes you positive or negative for certain blood types. This, real quickly, is one more marker protein. So that plus or minus represents the presence or absence of one more antigen to your blood cells. So if you're positive for any of these four blood types, that means you have a third antigen possibility, that third marker protein. So it's the same scenario. If you have it, then you're not going to develop the antibodies against it. So that means if you're positive, you can receive from positive or negative because there's not going to be a new antigen that poses a threat. However, if you're negative, that means you don't have that third marker protein. You don't have that third antigen. So you cannot receive, if you're negative, from anyone who's positive because they introduce a new antigen and therefore your body will attack it. So to be more specific, the universal recipient is actually AB positive and the universal donor is actually O negative because someone who's AB positive actually has all three of the marker proteins, including that third one, the rhesus factor. If someone was O negative, then they don't have any of the three of those proteins. So that last positive and negative isn't really anything you need to memorize, but just in case you were curious and you wanted to know that. So if you're O positive, for example, you can receive from only other O positives or O negative. If you are O negative, you can only receive from O negative. So O negative is the blood type that that blood banks and hospitals really want as much as possible because O negative officially is the one that, because it doesn't have any of the three marker proteins, can give to any other human blood type. Any other human can receive O negative blood without any complications. If you're AB positive, you pretty much hit the jackpot in terms of any situation where you needed blood, which of course isn't hitting the jackpot, but if you needed blood and you were AB positive, you can receive from any human blood that's out there because you have all three marker proteins and therefore you won't attack any combination of the three. Now the last thing we're going to talk about here are genotypes of these blood types. This is another example of codominance, and this is an introduction of multiple alleles. We always deal with multiple in terms of two, but if I say we're dealing with multiple alleles, that means more than the two that you normally have. In this case, blood type has three possible alleles for that particular gene. That particular trait has three possible allele combinations. There are two possible dominants, codominant alleles, and then there is one recessive. So here's how we're going to break it down. We have three possibilities for this genotype. We're going to use the letter I in this case. We use the letter I to represent something called immunoglobulin. It's the basic protein that's found in your blood cells and your immune system. The I is what we're going to use as the allele. So instead of there just being one capital and one lowercase, there are actually two possible dominant alleles, and then there's one possible recessive. So there's an IA that's considered the dominant for the A antigen. There's the IB that's the dominant for the B antigen, and then there's the recessive I, which represents no new antigen. So the IA represents the A antigen. The IB represents the B antigen. And the recessive I represents no new antigens. Can't spell today, sorry. Alright, so again, because you have two sets of DNA, you have two of these alleles, one from mom and one from dad. So everybody has two I's that's some combination of these. Now, all of the different possibilities give you all the different possible blood types. So if I'm talking about type A blood, that means I have the A antigen, but I don't have the B antigen. So there are actually two possible genotypes here that will show for type A blood without the B. You can either be homozygous for type A, which means you're IAIA, homozygous IA. In this case, all of your marker protein, all of your red blood cells show for the A antigen and only the A antigen. There's no B present here. Now another way to show for this genotype, hopefully you can guess this, is to show for the dominant allele with a recessive. Because in this case, this is the dominant showing and masking that recessive. So again, this individual will also have only the A antigen without any B's. So these are two ways to show for the type A genotype. The type B genotype means you only have the IB and no IAs, right? So in this case, you can be IB-IB, homozygous IB, or, just like the other one, you can be heterozygous IB and still show for type B blood because you have the B marker proteins or the B antigens and no representations of the A. Now type AB blood, there's only one possible genotype because you have to have one from each parent. Each of the two antigens have to be represented, so that means you have to have one of each. So you have to be IAIB. And remember, IBIA is the same thing, so always put the first letter of the alphabet before the second. So IAIB is the only possible genotype to show for both the A and the B antigens in all of your red blood cells. This is the codominant representation. This is the one that we consider codominant because both of the dominant alleles are individually represented. There's no mixing or anything like that. And then the final possibility to be type O means both of your I's have to show for the lack of either the A or the B antigen. You can't have either one of the two dominant alleles, because if you did, then you would therefore not be type O. You would have one of the other alleles. So the only genotype to show for type O blood would be homozygous recessive. So if you understand this basic scenario here about the different genotypes, then you can really get into some interesting questions about two parents that have their blood types and combine them into crosses. I've said over and over how a lot of these different scenarios that I say are oversimplified, and really your traits deal with multiple parts of your DNA or considered multigenic, and you really can't break it down this easily into just one particular trait and two alleles. But blood type is actually an exception. This is very straightforward. You have your blood type because of this very specific part of a very specific chromosome. So you can actually do some crosses here that are 100% accurate. You can tell what the probability of two people combining and producing a child. You can tell me exact probabilities of what their blood types would be. Let's do some practice here. Let's say I have a parent who's homozygous type A crossed with a type O parent. So parent 1 is homozygous IA crossed with a parent that's type O. So if I do this cross, the first parent, again I separate out the gametes. The first parent will always give an IA because both of the alleles are the same. The second parent, again, will always give the same allele. This time they happen to both be recessive. 100% of the offspring will show for this genotype, heterozygous IA. So what blood type would all of the offspring be? 100% type A blood. So 100% of the offspring here will be type A, even though one parent's type A, the other parent's type O. So one interesting cross here. Let me give you a cross representing my family. Okay, my mother was type A, my father was type B. I say was, they are. One parent is type A, the other parent's type B. However, I'm type O. Tell me if this is possible. I have one parent that's type A, one parent that's type B, and I'm type O. Is something wrong here? Do I need to go back and check my birth records and make sure that these are actually my two biological parents? Or is there a way that a type A parent and a type B parent can actually produce a type O child? These are my actual parents, so it is possible. Let's think about how that's possible. I need to be able to have both parents give me a recessive I, which means a recessive I had to come from each of my parents. My mother had to give a recessive I, as did my father. Now if my mother has one recessive I, but she's also type A, that means that the other I has to represent that A. So this has to be my mother's genotype. And my father is the same scenario, just with the B. He's type B, which means the other allele has to represent the B. So he has to be heterozygous for that type B blood. So this is the only way that I could have been produced the way I was. My mother and father both had to be heterozygous for their alleles, which means it is possible for me to be produced. And here I am today to prove it. So my parents are what you see here, and I am homozygous recessive. I am type O blood. Now my brother is type B. Is that possible? Yeah. This cross actually can produce any one of the four blood types, and it's an equal probability of producing any one of the four. So even though one parent's type A and one parent's type B, every one of the blood types is possible here. Here's my AB. Here's my B. Here's my brother, by the way. Here's my A. So every one of the four blood types has an equal opportunity to be shown if you have two heterozygous parents. One is heterozygous A, the other is heterozygous B. So again, you can do a lot more practice here and kind of do some crosses with it. Here's one question I can ask you. If one of the parents is type AB, is there any way a type AB parent can have a child that's type O? It's not possible, right? Not possible at all. And this is one of the ways that hospitals and police stations and things like detectives do paternity checks, and they figure out if someone's the father. They look at blood type here and see if it's even a possibility. If someone's type AB blood, remember that means that they're IA, IB. So 100% of their offspring will get at least one dominant I from that parent. It doesn't matter what the other parent is. That first parent will always give a dominant allele, which means there is no way an offspring can have two recessive I's. If one parent's type AB, then that means it's not possible to produce a type O child. The other 3 blood types are possible, but they depend on the other parent. Okay, so your blood types are all based on your genotypic combinations, the 2 alleles that you receive from your parents. Just like any other trait in your body is based on 2 alleles, one from mom, one from dad, and how they combine together. Thanks a lot, guys. Take care.